Welcome to all dear students in Gokul Global University. Today we discuss about topic is UV spectroscopy, instrumentation part 2 and the part 4 of the UV spectroscopy lecture series. So we also discuss about the instrumentation in the last part 3, UV spectroscopy part 3. So you start from the prism. The prism is made from glass, quartz or fused silica. So prism most often prepared from the glass material, quartz or the fused silica. Quartz or fused silica is the choice of material of UV spectrum. That means in the UV spectra methods, quartz and or the fused silica is the mainly choice in the material of UV spectrum. When white light is passed through glass prism, dispersion of polychromatic light in rainbow occurs like red, green, yellow, indigo blue, blue, violet, etc. colors. Now, by rotation of the prism, different wavelengths of the spectrum can be made to pass through in exit slit on the sample cell. The effective wavelength depends on the dispersive power of prism material and the optical angle of the prism. So again, the effective wavelength depends on which factors? The factor is dispersive power of prism material and the optical angle of the prism. Now the next, here the system described of the prism here. White light passes through the prism and the dispersed in 7 color: red, orange, yellow, green, blue, indigo, violet. There are two types of mounting in an instrument. One is called common type, which has an optical angle of 60 degree and it's adjusted such that on rotation the emerging light is allowed to fall on exit slit. So this is a cornu type and it's have a 60 degree and it's fall on exit slit. So when the light passing through this type of prism, it's refractive and the fall on the exit slit. The second is the other type is called litro type, which has optical angle 30 degree and its one surface is illuminated with reflected light back to pass through prism and to emerge on the same side of the light source. It is light doesn't pass through the prism on other side. That means this type of the prism does not passing the any light from inside of on material. It also reflect the radiance to the light source. Gratings are most effective. Gratings are most effective one in converting a polychromatic light to monochromatic light. As a resolution of plus minus 0.1 normal nanometer could be achieved by using gratings. They are commonly used in spectrophotometers. So chromato gratings are more important in the UV spectroscopy. There are two types of gratings. The first is diffraction gratings and the second is transmission gratings. Now the diffraction gratings. More refined dispersion of light is obtained by means of diffraction gratings. This consists of large number of parallel lines, grooves about 15,000 to 30,000 per inch is ruled on highly polished surface of aluminum. That's so we call the gratings to the aluminized surface. These gratings are replica made from master gratings by coating the original master grating with epoxy resin and are removed after setting. So these gratings are prepared based on master grating by the epoxy resin and are removed after setting. To make the surface reflective, a deposit of aluminum is made on the surface in order to minimize to greater amounts of scattered radiation and appearance of unwanted radiation of other spectral orders. The gratings are blazed to concentrate the radiation into a single order. So the surface of gratings are prepared by the aluminum metal and the surface is covered by the aluminum, so it's called as aluminized, in order to minimize to greater amount of scatter radiation and the unwanted radiation. So for this problem, the gratings are blazed to concentrate the radiation into a single order. Now the second is transmission gratings. It is similar to diffraction grating, but refraction takes place instead of reflection. So refraction produces reinforcement. This occurs when radiation transmitted through grating reinforces with the partially refracted radiation. So it is similar to the diffraction grating, and it's not a refractive, it's a reflective, and it's a reinforcement process consuming. So it's a very useful for the UV spectroscopy. Now the advantages of the gratings. Grating gives higher and linear dispersions compared to prism monochromator. Can be used over wide wavelength ranges. Gratings can be constructed with material like aluminum, which is resistant to atmospheric moisture, provide light of narrow wavelength, no loss of energy due to absorption. So there are 5 advantages of the grating system. The first is grating is very linear or the higher in comparison of the prism. It's used for the wide wavelength. It's contain a material like aluminum which is not oxidized by the any type of the atmosphere instantly. Then it's resistant to atmospheric moisture. It's provide also light of narrow wavelength, and there is no any type of the loss of energy due to absorption. Now we also compare the prism and grating with each other. So first the preparation made of the prism is made of the prism is glass for the use for visible, quartz and fused silica useful for the UV spectroscopy, and alkali halide useful for the IR spectroscopy. And in the view of grating, grooved on a highly polished surface like aluminium. Now the working principle of the both. The working principle of the prism is angle of incidence, and the working principle of the grating is law of diffraction. So the benefit and losses of the prism and grating, merits and demerits. Prism gives non-linear dispersion, hence no overlap of spatial order. It can't be used over consideration wavelength ranges. Prism are not sturdy and long lasting. So prism have to more carefully handle. Prism gives non-linear dispersion, hence no overlap of spatial order. That means prisms not give a particular light, its dispersion in the very different types of the lights. It can't be used over consideration wavelength ranges, so it's used for the particular wavelengths. And the merits and demerits of the grating. Grating gives linear dispersion, hence overlap of spectral order. It can be used over considerable wavelength ranges. Grating are sturdy and long lasting. So now we discuss about the sample cell, sample holders or the cuvettes. The cells or cuvettes are used for handling liquid samples. The cell may either be rectangular or cylindrical in nature. For study in UV region, the cells are prepared from quartz or fused silica, whereas color corrected fused glass is used for visible region. The surface of absorption cells must be kept scrupulously clean. No fingerprints or blotches should be present on cells. Cleaning is carried out washing with distilled water or Whitehall acetone. So the sample holder or the cuvettes are used for the handling liquid samples. It's have to vary cleaning parameters or it's have to carefully maintain, and it's made from the quartz or fused silica. This is a picture of the sample holder. This also known as a cuvette, and it's made from the quartz. Detectors. The detectors in the UV spectroscopy is very useful. So detectors' main work is detect the data is passing through the sample cell or the reference cell in view of radiation light. So here detectors, device which converts light energy into electrical signals that are displayed on readout devices. The transmitted radiation falls on the detectors, which determine the intensity of radiation absorbed by sample. The following types of detectors are employed in instrumentation of absorption spectrophotometer. There are three types of detectors. The first is barrier layer cell, photovoltaic cell. The second is phototubes or the photoemissive tubes, and the third is photomultiplier tube. So there are some requirements for the ideal detector, because the detector is the main important part of the UV spectroscopy or the spectrum spectroscopy systems, because of when the detector is work perfectly, then it gives perfectly data to the amplifier, and amplifier gives the data perfectly to the recorder, and recorder give the perfectly spectra of the any type of the data. So requirements of an ideal detector: the first is it should give quantitative response, it should have high sensitivity and low noise level, it should have a short response time, it should provide signal or response quantitative to wide spectrum of radiation received. So the first is it's one have to give a quantitative response, high sensitivity and low noise level, because the high noise is defect the accuracy of the spectra. It should have a short response time, because the detector have highly responded, then it saves our time during the practical, during the instrumentation work, during the spectral work. It should provide signal or response quantitative to wide spectrum of radiation received. Here the first is barrier layer cell, photovoltaic cell. The detector has a thin film metallic layer coated with silver or gold and acts as an electrode. So here electrode is used a metal of silver or the gold. It also has a metal base plate which acts as another electrode. These two layers are separated by a semiconductor layer of selenium. So here in the diagram you also see in the bottom it is a base plate like iron, and on this plate the second layer is selenium layer, and on third surface transparent layer, and the all of the above collector ring which is work as a anode. So the when light radiation falls on selenium layer, electrons becomes mobile and are taken up by transparent metal layer. This creates a potential difference between two electrodes and causes the flow of current. When it is connected to galvanometer, a flow of current observed which is proportional to the intensity and wavelength of light falling on it. So here diagram of the phototube